My name is Siniša Bikić, and first of all, I would like to say that I'm honored to be invited here at this very prestigious conference, and I'm also honored to be chairperson of this session. So I will kindly ask Professor Damian Kelts to present the paper with title Frost Prevention in Vineyards Powered by Renewable Energy. The floor is yours.
Thank you very much. So first of all, I hope you enjoyed your lunch. I would like to greet you all in the name of University of Maribor, Faculty of Agriculture and Life Sciences, and we are coming from chair of Biosystem Engineering. Thank you all for coming on such a nice congress event, and thanks to organizers for organizing it. So I would like to say something about frost prevention in vineyards that are powered by renewable energy. So we all knew that these recent extreme climate events across the globe highlighted the critical need for innovative frost protection strategies in agriculture. Frost can severely reduce crop yields, especially when temperatures dip during sensitive growth phases. So in answer to this, we designed a heating wire system and heating bulbs powered by a low power electrical setup and a small Darwin wind turbine with a solar panel. Also, a gravity battery system was developed to show an alternative method for storing renewable energy. So the materials for this performance, we used a network of some specialized sensors. Especially, we collected this data with IoT sensors, which works on LoRaWAN network. With these sensors, we triggered on and off the heating wire. So these sensors are here as a picture, as a photo. Presented. For a heating wire, we made 3D printed clips, and to secure the wire exactly to the plants, the wire was stretched 60 centimeters above ground, drawing only 20 watts per 1 meter. The wire offered energy sufficient supply heat to tissues. Here are also these sensors photos from vineyard. So another version we also made of this heating wire, which draws 25 watts per meter, the heating power. So the newer system also features the controller. So to enable user easily turning the system on off during climatic events. About energy solutions, we also explore ways to harvest and store this energy on site. So in vineyard, for example, that's why we made a hybrid Darwin wind turbine. This system is primarily designed to harness wind power and to convert it into electrical energy. Photovoltaic was also included to harvest solar energy. The materials for this Darwin wind turbine, we used 1 kilogram of PLA white plastic for 3D printing, the housing 3 solar panels, and a wind turbine to convert wind energy into electricity. And we also included 20 ampere hours rechargeable battery to store the generated energy and to provide consistent power output. So the photo from Oshart, you can see this hybrid Darwin wind turbine that was 3D printed. And so some another energy storage solutions. We investigated also a gravitational hydro battery. So on a farm, it is a solution for running automatic heating systems. The prototype stores power by pumping water to an elevated reservoir. It is, in our case, it was 4-meter high distance. And later generates electricity by passing the water through the micro hydro turbine into a hydro generator. So the experimental setup involved with water storage tanks. I will show a photo a little bit later. A water pump to move water to the upper tank and a hydro generator. A concept can be produced based on available infrastructure or even on a landscape at a farm, for example. So this is one low-cost example of storing the water, and it was only 4-meter high. The difference between two storages. So the results for a heating wire, we can see that the unheated row is registers the lowest temperatures, often dipping below freezing. So it is on a blue color. On orange, you have heated row. It is heated row is performing much better temperatures, and we measure also a direct cable temperature. This is a green line, but this temperature is very high. So because the sensor was on the wire. So the data clearly showed that the temperature directly on the wire remains higher than the other points, demonstrating a stable heating effect. For optimal frost prevention, it would be beneficial to place the wire position closer to important areas, such as wrapping it along branches or directly under sensitive crop zones. Then we used also heating bulbs. The photo of these bulbs is on the right side. It was Philips bulbs, 180 watts per one bulb. And on this figure, we see that both unheated rows at 40 and 140 centimeters remained significantly cooler through the observation period. Yeah, the difference is on this figure. That means that experiments show clear differences in temperature across the heated and unheated rows. Air bulbs provide localized heating effect. For frost prevention in sensitive crops, the bulbs must be positioned as close to the target areas. And infrared radiation requires the use of multiple bulbs or careful placement to ensure effective protection across the entire area. And there are some results of mobile wind turbine. So our experiment with the ventilator system demonstrated that directing air from elevated positions had a minimum effect on temperatures at the ground level. So the fan system did not cause significant changes in temperature. That is confirming to the statement also of Mr. Tadic that the performance of wind machine is low. We would recommend increasing the system height from 10 to 20 meters to better utilize the observed temperature gradient. So hybrid Darwin wind turbine system shows promise for a low power application in agriculture. System could evolve into a sustainable and cost-effective solutions for targeted use. And the similar figures of Mohammed provide us that further research would be beneficial. And now conclusions. Our field test of the heating wire system did not yield the level of success we expected. Due to its low cost and ease of implementation, we plan to continue research on this area. We conducted that using ER light bulbs provided successful in plant protection. They are easy to install, trigger, and provide a way to prevent frost. Disadvantage is their high power consumption and energy that is not transferred into head of the plants, but in the also in the surrounding area. So in future, we could investigate methods for capturing the excess heat. It may be possible to redirect this loss thermal energy towards areas of protection. And this could improve the overall efficiency. And the ventilator system experiments shows that the fan system did not significantly impact ground or near ground microclimatic conditions. The hybrid Darwin wind turbine system present cost-effective energy solution for localized frost protection. Future improvement, such as integrated high-capacity batteries, for example, or modular configuration using more efficient technologies, could significantly expand its potential. So thank you for listening to the presentation. Thank you for your attention.
Thank you very much, Professor Kelz, for this very interesting presentation. So do you have any question related to this presentation? Okay. If you haven't any question, then I will kindly ask Professor Rafat Al-Afif from Boku University, Vienna, to present us the results of research titled Challenges and Solutions in Biogas Production from Food Processing Waste, a case study. Professor Rafat, the floor is yours.
But we need to go to the presentation modes.
There is.
Already, huh? Hello, everybody. So first of all, I would like to thank the organizing committee for giving me this opportunity to talk with you today. Thank you very much. And I wish to welcome you with this presentation today. I'm going to talking about the topic as a key questions. I'm talking about today the problems and the challenge behind building the biogas plants or using this technology, and try to give some solution or some suggestion. For sure, I will not cover everything because of limiting the time. As you know, the sustainable production of biomass fuel, it's like recycling the carbon dioxide atmosphere and rich the carbon nutrient, is in the line of the United Nations thematic to protect the environment and also contribute to sustainable development goal to produce affordable and clean energy. So biogas production from the anaerobic digestion of no? So always we expect some technical things. So yeah, it moves. So actually biogas production from anaerobic digestion of biomass, so or organic material, to produce it is can say renewable and clean energy, and we can consider it as a transmission fuel to fully decarbonize our energy consumption or supply as well. Okay, why? Because it make potential to reduce the waste problems, and also to the benefit for the environment. When we're talking about the environment, we're talking about the reduction of carbon dioxide. So if we substitute the fossil fuel with the biogas technology, we expect to reduce 2 kilograms carbon dioxide per metric cubic biogas. This is really sufficient, because actually if we need to produce kilowatt hour, because metric cubic of biogas equal one of the has a energy or heating value as kilowatt hours, so it means 10 kilowatt hours, then this the significant reduction of carbon dioxide. As I promised, I will not go to the details today, but I'm just focusing on the problem and the challenge and the solution as well. So one of the question, if this technology is good, why we don't use it, for example, in the wide side for the countries? For sure, in Austria, we do that. And we have a lot of biogas plants. And I will try to review today some of this policy to increase, to develop this technology. But one of the, we have many challenges behind this. The first one, this is the inhibition of biogas production. So if we're talking, no, I need to, because I need to go to the from a slide to others. If I go to the presentation mode here. I understood, but I make something nice to go from one to others. So if I had a presentation mode that I'm able to link. Sorry for this, but we can continue that. If you put this in the presentation mode, then I think now. I will think yes. Okay. Then I will continue. Sorry for this interruptions, but I'm talking about the challenge. It's one of the challenge actually inhibiting of the biogas production. So so I see it is not possible. Why? I don't know. So I will go to the, because I can't, if I put it, I have to go to the.
There's a
Okay.
other side for this, or it's just other slide.
No, I need to come to here. To link. Okay. Okay, actually, I will continue like conventional. So if we're talking about the inhibition of the biogas production, one behind this, the structure of biomass. Yeah, so the biomass consists of mainly cellulose, hemicellulose, and lignin. And lignin most likely surrounding the cellulose and prevent the bacteria to break down the bonds. And then this one of the challenge to produce biogas, because we use this anaerobic digestion, we use the bacteria for this, okay? And other challenge, so if we have some heavy material or antibiotic material like phenol, for example, if we need to use this biogas technology from food industry like olive oil production, so the waste from olive meal, so it means has some concentration of phenol, and accumulating this phenol could be poisons the bacteria and inhibit the process of biogas production. So actually, this one of the challenge. Other challenge, the technical challenge. When we're talking about technical challenge, we're talking about the purification of the biogas, because biogas consists of carbon dioxide and methane as well, and hydrosulfide. And we need to extract or to get rid of the carbon dioxide and hydrogen sulfide, because these don't have any potential or energy potential there. And hydrosulfide has a negative effect to the corrosion of the pipes and so on. So actually, this one a challenge. And if we're thinking about using this, not only the biogas to produce electricity through combined and power unit, nevertheless, with the fuel cell also is the source to get the electricity. And in this case, we have to make the reforming of the biogas, the methane, to produce hydrogen, and then use the hydrogen in the fuel cell to produce electricity. And this costs also money, and this is the big challenge. Other things, so if you need to select the fuel cell which really fit to this methane or biogas production directly. I will talk about this later. Actually, other challenge, this economic challenge, and this is what I see it's important, because the high cost of building such technology, it means effect to the levelized cost of energy. It means how many euros cost the 1 kilowatt hour, because we need electricity, and still this high. If we look to the specific investment cost, this is about 3,000 euro per kilowatt. So it means if we need to build 1 megawatt, for example, this is about 3 million euros. Yeah. So this is like to think about. And also, you can see here specific investment cost for reactor, for combined and power unit, and for the feeding system, and so on. It means now I don't need to go in details. I will keep this to the questions. I promise to finish and give time for the questions. Actually, another challenge, this is the policy challenge. It means it's not enough to we need the support from the government. It's important, okay, to, for example, to give this technology free from taxes, okay, because this affected to the price. And we need to the support from the government, because the kilowatt hour, comparing with the kilowatt hour, the cost of kilowatt hour from biogas production compared with other sources, it's still high. But because if this keep the environment and many of benefit I talk about, we need the support. Okay, if we're talking about the solution, the first one, we're talking about the optimizing of the biogas production process. Okay, and here the optimizing of biogas production could be in many strategies. So first, improve the biogas production through sustainable pretreatment, okay, or substrate pretreatment. As I mentioned, this is in the mechanical pretreatment or the chemical or supercritical carbon dioxide pretreatment. We hydrolyze the lignin and expect more biogas production. Here I have some publication. We're using here the carbon dioxide, supercritical carbon dioxide pretreatment, and this affected really about the quality, quantity of biogas and shorter the hydropretension time, and this affected in the investment cost. Why? Because the volume of the reactor we can calculate, simply multiply the organic loading rate, multiply with the time need to complete the biodegradation. This is the hydrogenation time. If we shorter the hydrogenation time, it means we decrease the infrastructure costs also. Okay, this is also another study which confirm these things. Yeah. So other things. So we have the environmental conditions. What I mean for the environmental condition, many of the biogas plants, for example, they build well, yeah, but they lack of knowledge what the optimal conditions need to grow up the bacteria and then to make this process more efficient. For example, the temperature. So it means if we're thinking about the mesophilic temperature, I would say 35 plus minus 2 degrees, but not between 40 and 50 degrees because the bacteria never adapted in this. And I met with the Professor Sinisha Pikic, one of the director of the biogas plant in Serbia, and they adjust this in 42 or 43 degrees. Yeah, I'm wondering, I told him really, because the outcome, it's low, and the problem, it's very clear, very simple, we can lose it, service, but we need just to know about the condition. I am not here to talk about all the condition, just this one example. Okay, if we're talking about additive, also if we add, for example, enzymes, also this is faster the hydrolysis and to produce biogas. Nutrient supply, also we can supply glycerin because this is also effect in the quality, the concentration of methane in biogas because the methane is the energy holder. So other things we can talk about the optimizing of the performance. It means, as I told, the hydroretention time. If we shorten the hydroretention time, this affects positively to the cost. Okay, and also the organic loading rate, because if we put more substrate in the digester, what we expected the pH will be dropped down because the fatty acid increased. This also one of the facts can affect it. So we need to be sure what really the organic loading rate and hydrogenation time, also the yield or the rate of biogas production. And this is metric cubic per day. If this less than 1.5, it means we have problem. We have to search what really, how we can solve this problem. Okay, so actually other things we can think about that developing the new technology. So I think this is what we are seeking for all the time, to develop the new technology. Okay, so here we thinking about demonstration of the innovative technology for improving energy output. What you see here, so it means we have the steps behind digestion of the organic material under from the raw material to get the biogas and the sludge. Okay, I am not going to talk about this now, but I'm going to talk about how we can develop the technology. And from the beginning, we talking about the feeding system. We need the good, so improve the of the feeding system. And also we, if we're going to talk about the digester itself, it means the early warning system. Yeah, it's very important to control the condition. And now we're going to the artificial intelligence. It means now the next step that the digester can adjust itself. It means can decrease the organic loading rate to keep, for example, the optimal pH about 6.3, for example. Yeah, so it means or the temperature increase and decrease, so automatically. So we're seeking for these things now, and early warning. If we have, for example, the rate of biogas less than 1.5, then we have problem. Yeah, we have some alarm. It's also, this is, and if we're talking about the last stage to produce electricity, this we can do through the combined heat and power unit. And here we suggest to use the organic ranking cycle. And here we can, in the not high temperature to produce electricity, not like the water cycle, for example. Also, we need to think about heat exchanger. It means the heat we have, we can use to heat the digester, or we can drying the sludge or something like that. Okay, so to also the advanced purification and reforming of methane. This is we need to think about also using some solid oxidized fuel cell that we can use directly the methane to produce electricity through the fuel cell. Actually, other things. So we talking about maybe. Okay, we develop the proprietary biogas technology. It means every country has own system. We need to modeling the system based on the need and the country. Also, it's important to think about the how to control the system. And in this way, how to control the system, I think. Okay. So it's not here. I'm sorry because of these technical things, but okay, that's I told you we in Austria, for example, before the farmer or the company build the biogas plant, should make a contract with the scientific research or the university that they every month go and take samples from the biogas and from the sludge, and they ensure the sustainability of the work. And they have to pay money for this. It's no way because they need to ensure the sustainability and that everything going well. So other things to integrate with the other renewable energy. That's what we now working in our institute, and to make the hybrid system. It means to combine the biogas production with the thermal conversion, with the solar, with the wind together, and make modeling for this. This also very efficient to reduce the cost of the kilowatt hour per, yeah. So the policy support, I will suggest is different from country to country, but I will expect that the country will be support the biogas through this low bureaucratic things to and make life easier for the investment. So, and by implementation, this solution, biogas can become a more important contributor to the renewable energy mix. And this is some publication in hybrid system. Recently, we do projects with Tunisian and with Jordan in Middle East, and now one running a project with Uganda in Macquarie University also. I would like to thank you very much for good listening. Thank you.
Thank you very much, Professor Rafat, for this really interesting presentation and valuable information you shared with us. So do you have any question for Professor Rafat? Here we are.
Thank you very much for your very interesting talk. And I was wondering, right now in my hometown in Madrid, there is a big population movement against the building of one of these biogas plants. And I wonder if there are concerns about the smells and the other inconveniences are real, or they are just imaginations. I mean, is this concern, is this a problem, the emission of other inconvenient gases from these installations?
Thank you very much for this, because I appreciate this question, because I really forget to talk about social challenge. Yeah, increase the awareness about the benefits for something. It's important. So actually, this is anaerobic digestion. It means without oxygen, and all the diseases will be killed. And then we don't have methane. So it means we don't have ammonia, yeah, in the digested, because this is convert to ammoniac with the water. Yeah, so it means we don't have smell. So it is not the, but so the one of the aim of the biogas production to reduce the waste deposit problem. It means the substrate, the manure smell, yeah, the silage also as well. But using this in the right time, yeah, and this waste management, also this is another thing you need to think about. But I think it is not the it's not the reason that they brought it. Just I expect to tell me about the cost. I agree with you until now. Yeah, but what I suggested here for sure reduce the cost of the kilowatt hour production. Yeah.
Thank you very much for answering. Do you have more questions? Okay, if you haven't got any question, then I will now invite Professor Balázs Bancarovsky. He will present results of research titled From Subconscious Reactions to Consumer Preferences, a multimodal sensory study investigating taste perception using galvanic skin response and facial expression analysis. Professor, the floor is yours.
Thank you very much. Dear audience, dear everyone, please let me welcome you today. My name is Balázs Bancarovsky, and first let me tell you it's a very big honor and a privilege to be here with you, and for the opportunity to show us your, our study, which is deals with some multimodal projects, I would say. So first, you may can ask, what is a sensory guy doing here? Because we are in a very BiosysFoodEng conference. We have a very special topics throughout the whole food science and food technology. But in my opinion, I think the sensory science is the absolutely first start of every field of science, and absolutely should be the end of every field of science, because we are dealing with customers. We have to deal with a lot of people with different requirements, different needs. And just like you can see in this graph, the market of this sensory science is increasing a lot. If you can see in the last column, the market size can be grown until 140 million dollars, but is a really, really huge market. But what we have besides the money, we have so many good new technologies. For example, biometrical sensors, or for example, modified environments in virtual reality, augmented reality, or even artificial intelligence. So we have the technology, we have so many consumers, but we have to do something with that. And of course, I don't have to mention here, because you already know we have so many new challenges of this century, especially for the sustainability, the resource management, and food waste as well. Just a very short information: in public catering in Hungary, in every year there are a really big food waste, especially 50 million kilograms for each year, which is huge money, huge amount of wasted money, and so many people can eat that kind of portion what we are going to waste directly. Especially, and only one more information about this, the most of the situations, the soups are creating this kind of waste. Every second portion is go directly to the waste. But why? It can be a really good question, and this is where the sensory science joins here. Because, just like I mentioned before, we have so many biometrical, biosensorial opportunities. For example, in galvanic skin response, or in short, GSR. You may know this technology. It works like a polygraph in the criminal movies. It's like works with skin conductance, and we are transported it to skin response with the sweating gland of what's created in a station corneum, because during the cells and during live situations, for example, I'm sweating, and that sweating creates so many metallic ionic movements in the sweating glands, and that during this moving, that creates this electrode potential difference, and that's what can be measured with summary sensors, for example, silver chloride sensors here. And that we can transport it to skin response specialities. We have so many precise devices for that. For example, what we use, basically, it has 127 data points that been saved every second. So we have very big data as well. But if we want to analyze more these kind of signals, we have to separate we have to separate these kind of signals. One, it's linked to the brain activity, or we call it aerosol. The called HCR. And the second one, just like in blue here, this is the HCl, what is the casual mood, I would say. But during some exam periods or exam situations, we have this big brain activity, and it has caused some intensive things during the body. But what we have more, we have the face reader, what is basically a camera, I would say, but in so many cases, it has so many more detail about that. For example, there is a supplier called Noldus. We had what deal with a very big data set about each participants with different demographic, with different age and different nationality informations, and they have saved it and use it for modeling. But what they are saving from that, some key points of the humans' faces, especially for nose, eyes, or even eyebrows or mouths, and you can evaluate it during the whole process, but it's based on what kind of products you have. In the bottom slide here, I just tried to calibrate myself, and luckily I'm a good model at this point. So Just like I mentioned, it measures some emotions based on these mapping technologies, especially the basic emotions, for example, happy, sad, etc., and also the neutral informations. But we have, from the last century, some, how should I call it, schedules, especially if there is a special one, what's called positive and negative affect schedule, or in short, PANAS. PANAS has two big type of emotion, what is already described here. There is some positive and negative effects, and if we want to add them together, you can rate it from 1 to 5. You will get this PANAS score, and you will get some informations about your emotions moving here. Just like, for example, our model here, but for us, it's really hard to understand his emotions right now. So we made some experiments which deal some facial and psychological expressions to comparison between them, and can we predict one to the other, and how is there any tendencies, and if they are, what are them? So we recruited 40 participants in our university, Hungarian Agriculture and Life Sciences, and we asked them to first fill a PANAS questionnaire. How's their actual feeling? What's their opinion? What's their actual feeling? And after that, we asked them to drink and evaluate two different types of liquids. The first is the vegetable soup, which has different attributes based on the salty and umami attributes, and we also ask them to taste and evaluate some lemonade samples which has some difference between the content of sugar or, for example, citric acids. And after, during the measurement, we use this galvanic skin response, and also we measure this skin response, and also the heart rate, and also we measured after the experiment their PANAS results. For the evaluation, we use so many devices and so many softwares. For example, we used this kind of face reader for understanding the facial emotions. We used Shimmer 3 Plus governance skin response meter, and for the evaluation, the statistical evaluation, we use Redstate and XL-Stat. But what was our main result? First, we want to understand how they evaluate the different attributes and how they affect for the overall liking. First, let's see the vegetable soup informations. Here you can see the most liked product. It get 6 point out of 10. Was that product which has more umami concentration, but it's a very interesting because okay, they were served in a warm temperature, so the temperature cannot be a very big effect for this. But as you can see here, the most penalized attribute was the salty, but that sample doesn't contain any salt. So in the case of these consumers, they cannot separate the taste of the umami, and they cannot from the salty. And really interesting, the lowest score, overall liking scores, went to that sample which has combined, I would say, intense attributes. So it also contains umami and salty as there, and as you can see, they weighted it because they thought it's a very huge amount of these kind of additives are in the product. So that means if you want to imagine, it's like 70.5% of each cup. In case of lemonades, here you can see the most liked liquid, I would say, or sample was that one which has got this increased lemon and sugar content, but they also found that it was a really sweet sample. And the most not popular, I would say, was the control sample, which has got only 10 or 15% of these kind of additives. But if we want to analyze it for them, and we want to get a concrete picture or a screen how they can be categorized, we can perform this internal preference mapping, but based on the correlation of principal component analysis. Here you can find it can be correlated in the left side of the slide. It can be correlated to the overall liking, and I have to mention that the first two eigenvalues can link to the opposite tastes. For example, in the vegetable soup, you can see the left side goes to the salty side, and the other side, I mean the F1 value other side, that goes to the umami taste. But and the second eigenvalue can go the intensity of these each product. In case of the lemonades, we can see the similar informations, but not with salty, but with sourness from the left to the right. I mean, left to the right, there is the information about the sourness and sweetness, and the intensity was the same ratio here. But if you want to analyze this PANAS score, what's again, it's self-declared informations about the participants' feeling. You can see here, there is a really big decreasing tendencies, especially what was really surprising for me. Also, the positive effect is also increasing, but it wasn't our goal. So we have to go more deeper. Why is it happen in here? So we created this, I would say, wonder, or if I want to be polite, I would say multiple factor analysis, where we use all of the PANAS attributes, all of the information what came from the face reader, and also from the product liking here, and we found some interesting informations. For example, if I want to make it more clear, you can see here the negative and positive PANAS has goes different, I would say, quadrants, but there are some miscommunication or misunderstanding, because for the distressed attributes, what they by the schedule, it would be positive feeling, it went to the negative side of this evaluation. But I also have to mention here the face reader results. For example, the sad went to the negative PANAS, but it's also good. But if you see here, the scared and angry information went to the positive side of the scale. So it's really hard to understand these kind of information and attributes of each participant. But if we want to cluster them, we can use this agglomerative hierarchical clustering method that's based with Euclidean distance and word method. We can find that we have 5 different clusters, but in the most of the cases, the overall, like in the also the demographical data and the big variance of the demographical data caused these kind of big numbers of the clusters. And yeah. So in conclusion, if they ask me, is there any correlation between psychological signals and facial emotional tendencies? If I want to say directly, only for PANAS questionnaire, it's no, not this point. But if we can complete that with other kind of experiments or experiment methods, for example, if you want to include some VR technologies or eye tracking, or even if we can use this GSR here, we can get some more results and more information about the participant. And this field, I mean, for this virtual reality and also for this eye tracking measurement, is a very new and unique, but you will hear some information later in the presentation of Hanan. So I want to keep here that attention there. So from my side, thank you very much for your kind attention. I would like to thank the organizers to invite us to here, and also I want to thank to our colleagues for your help and dedication during the whole process. Thank you very much.
Thank you very much, Professor Balas, for delivering this very interesting topic and presentation. Do you have any question for Professor Balas? Okay,
I hope you like my presentation.
surely. Thank you very much.
Thank you very much.
I will kindly ask Professor Hinek Rubik to present the results of research titled From Waste to Wealth, Circular Bioeconomy Pathways in Agri-Food Systems for Sustainable Global Development. Professor Rubik, the floor is yours.
Yeah, can you hear me? Oh, all right, perfect. It's my pleasure to be here. My name is Hinek Robik, and I'm coming from Czech University of Life Sciences in Prague, and specifically from Faculty of Tropical Agri-Sciences. And today I will try to tell you a bit about what we tend to do. And the title of the presentation is From Waste to Wealth, but specifically looking into circular bioeconomy pathways in agri-food systems linked with sustainable global development. At the beginning, I want to say that I'm really happy to be here. I'm also happy to have the conversation with a lot of you. And of course, I want to mention that you are very welcome to visit us in Czech University of Life Sciences and specifically in my faculty. But now let's go directly to the topic at hand. I always like to start with explaining what the problem is. And I would like to mention that we sort of live in a paradox. If you remember, one of the first presentations today in the morning was mentioning that we need to produce more food and that we don't produce enough. I would change or say differently. We produce more than enough, but the problem is that we are not able to deliver it to the people who need it. So we are producing more than enough to give or provide food for more than 10 billion people. On the other hand, we still have around 800 million people who are suffering from being undernourished. So, and that the main reason with that is that over 1/3 of all produced food is being wasted. And there is various reasons for that. A lot is linked with logistics, a lot is linked with production, processing, post-harvesting. A lot of the topics have been mentioned here today. But then also what has to be mentioned is that agriculture is an important sector giving us the source of life, but also contributing currently quite a lot to the global greenhouse emissions. So we have systems which are designed to sustain us, to feed us, but we have to rethink them a bit. And as most of the big challenges are systematic, this one is also very systematic, and therefore the solution also has to be more holistic. What we tend to do at our faculty, we focus a lot on something what we call tropical agroecology systems. That's something what we are also introducing to FAO and United Nations and other big partner organizations. But something what you are probably much more familiar with is bioeconomy approaches, and therefore I'll be more talking about bioeconomy or circular bioeconomy, or as previously it was known, circular economy, which is more or less built on 3 simple principles. So using natural biological resources, but also using them wisely. And that is an important part. Then being able to recover and valorize waste at every stage. And that's quite an important thing, to be able to always create higher added value products. It can be fertilizer, it can be higher added value food products, it can be bioplastics, it can be almost anything, as long as you are able to produce something and keep thinking about the closed loop systems, because that's the only way how we can ensure that our planet will be survivable for next few hundreds of years. So I also like to say that we have to always think about designing both for food as well as for function, and thinking about the soil health, thinking about the bioenergy we can produce, being able to link all that, also in terms of nutrient recycling, as well as working with biomaterials. Now let's have a look. Because something what I like to say is from waste to wealth. Some of those who I have talked with today, they were asking me what is my background, and I usually say waste to energy. But in reality, I like to say there is no waste. It's only how we think about it. Now let's have a look on a couple of examples. I will show you a couple of examples of what we have done in the past, and also to give you an opportunity how to think about problem solving, and that it can always go even from small solutions, which eventually will turn into something bigger. One example, what we do a lot in, especially in Southeast Asia and Africa, but also other regions, is building anaerobic digesters, or as was earlier mentioned, biogas plants, where we are turning usually food waste or manure into energy and into fertilizer. And sometimes we also link it with some other products. Last year, we started linking our systems also with aquaponic and ponds and feeding fish, so making it even more circular. Till today, me and my team, we have built over 1,000 biogas plants around the world. So it's not just some theory, it's something what we really do and what we know works. Another example, and again, it's also a relatively simple thing when we are thinking about food waste. So in Zambia, we have started implementing black soldier fly larvae to convert organic waste into high protein animal feed and organic fertilizer. This is also something which you can do on small scale as well as on large scale. And it always depends how you want to implement it. And the third example, which we are now also quite actively doing, is in Cambodia, Vietnam, Zambia, is working with biochar. I think a lot of you are familiar with biochar as well. It's something you can produce from agricultural residues, and it has very good effect on enhancing soil fertility, being able to sequestrate carbon, improving farmers' yields. In the past, what we, for example, also did is we developed new feeds for animals with added biochar to reduce methane emissions and keep their growth. So you have many directions in which you can go. And always, yeah, these are not just pilots, these are already proof of concepts which can be scaled up. I'll show you a couple of pictures how it looks, so you can imagine it. It can be very simple. For example, here you can see small-scale biogas plant. They are processing organic waste into energy. It can be small system from 10 cubic meters to 50 cubic meters. Most of them we built around 30 cubic meters. But then, on the other hand, we also built biogas plants which are a few hundred or around 2,000 cubic meters. So it's a simple system, which, for example, here you can see it's usually linked directly with the farmers. These are examples from Vietnam and Zambia from something what we have built during the last year. Here you can see, again, a simple example of black soldier flies. So it's something we do in large scale with large scale producers, but it's also something which we do with small scale producers. This is pictures from Kenya and Zambia, where we were introducing it to the local farmers, so they can also have a new system how to deal with food waste and have some additional income. And then this is from 3 weeks ago, another simple example of biochar production. So of course, we do a lot of research with biochar in our case, and we also work with large-scale biochar production. But we also want to be practical. So we also work with local farmers. This is in Cambodia, where we have been since this year. We started to introduce more of simple biochar production, which we are replicating throughout the country. And it's, again, something which can be introduced in a simple way. It can be replicated in all conditions. So you can have something for the large-scale producers, and you can also work with the small-scale producers. And both of the things are very, very important. On the other hand, for example, this is processing of palm oil effluent that's from Indonesia. And this was biogas plant, which was of a size of 4 football fields. So then it's sometimes of a very different dimension, as I was saying, from small scale up to large scale. Now, what we want to think about is the circular benefits. We always have to consider the economic, ecological, and social aspects. That means when we are thinking specifically about the farmers, it means to help them reduce their input costs and that then create new revenue streams and increase their resilience. Now, you have to also realize that, for example, if you are coming from Hungary, the soil has here some issues, but not as many issues as in some of the other regions of the world. So, for example, when we work in Western Zambia, it's very sandy soil. So more or less the soil is sand. So you need to realize that it's very difficult to work there, and we need to be able to get there the soil health into the soil, which more or less is really a sand. Yeah? So otherwise, when you try to fertilize such a sandy soil, everything is washed out. But if you increase the carbon nitrogen ratio within the soil, then it's able to sustain the nutrients for longer. Then, yeah, thinking about the ecological aspects, help to regenerate the soils. And also, in our case, what we always focus on is the social aspects, practically increasing new jobs in the area. This is something which we always try to do when we are coming to the new country. We work with all the levels. That means the ministry, the local farmers, the local partners. And through the new system, very often linked with some technology, we are also trying to create some new local jobs. Very often, most of those solutions can be and are very low tech. And that's perfectly fine because it's important to have it replicated massively. If you want something high tech, you are able to have it on one place and you need highly skilled people to operate it. But if you are able to implement something which is low tech, you are able to implement it very widely. And that's why we tend to work with what we like to call holistic approach. So we have to think about all the aspects which are related to it, and always thinking how to, for example, connect farmers with biorefinery cities, with composting cooperatives, markets, with protein startups, stuff like that. Also, what is practically, or what we can say from experience is that we always need multidisciplinary approach behind it. So for a lot of solutions, we need agronomists, engineers, economists, entrepreneurs. So we are able to provide some solid and robust solutions which we can then apply. And also the support of the policy. That is the reason I also established around 2 years ago Agri-Policy Lab within our institution, which is a multidisciplinary unit, which also helps us to help our partner countries to implement new policies. Because a lot of new technologies, you need to also link it with the policy. Yeah, now we have also the opportunity to think more about the digital tools and digital solutions to accelerate it even more. In our case, what we use a lot is different remote sensing approaches, usually to be able to identify and map different areas. For example, now we have a big projects where we are using remote sensing for mapping new areas for coffee growing in Colombia. and being able to predict or help them predict how they will be able to grow coffee in next several decades, because also the climate change is changing a lot of things. Similarly, it's possible to work with blockchains, especially linked with nutrient flows and waste valorizations. And another thing which is not that difficult is different mobile apps. So to help people in the real time to sort different problems. Now we have finalized and started testing a new app for which is called Wheat Plus, which is for detecting different wheats. And we are planning to start testing it next year in 2 African countries and see if it will be more applicable. It will be, again, open source software. But the mobile app, especially with the help and linking it with the AI detection of the weeds, it's not that difficult to develop these days. It took us less than a year, which is, you know, relatively short time when you think about it. And to mention also that it's good to think about a global south as a landscape of possibility, because very often we tend to forget that most of the global south is very uniquely positioned into circularity, with the abundant amount of biomass, with a huge amount of natural resources. And that applies for Asia, Africa, Latin America, which they have an amazing potential, and it just needs to be worked with it. I also like to remind to always try to work in the context-specific approach. That means that some of the solutions we have here, let's say in Europe or Central Europe, very often they does not necessarily apply in the other parts of the world. So we need to always add there the context-specific knowledge. And also, this is an experience we I would say learned the hard way, but it's easy then to replicate that. Most of the solutions which were replicated the best were community driven, and that's the reason why we very often work with the local communities, because once they have the ownership, then they are happy to work with it and replicate it further. And that's the best way, because you never can push innovation on someone. You need to create a space where people want the innovation and they believe it's theirs and they want to work with it further. Before, I've seen at some of the presentation the linkage with the SDGs, and that's good to always show. But at the end, it's actually linked with all of them. But if we would highlight a couple of them, it's SDG 2, 7, 12, and 13. But as I'm saying, it's always linked with all 17 SDGs. So to close it a bit is when we are thinking about turning waste into wealth, we always have to think about transforming how we think about it. And what I like to remind is we have to stop seeing waste as a burden and start always to see it as a resource. And also not to always think about producing maximum yield, but more about circular efficiency and systemic balance. So we need the yield, but we also need the balance, and we also need the whole loop to work. So to conclude, when you next time look at a pile of waste, don't ask yourself how to get rid of it, but think about what you can do with it. Because we are now at the crossroad. One path can lead us to more of resource depletion and inequality, or we can go more into the circular path, which can lead us more towards regeneration, innovation, and shared prosperity. So I hope that we'll be able to take the second path and turn waste into wealth. Thank you very much.
Thank you very much, Professor Rubik, for this really comprehensive presentation on the topic of conversion waste to wealth. So do you have any question for Professor Robic?
Feel free to ask, or feel free to reach to me later as well. Yes, please.
Okay, here we are.
Thank you very much for a very nice and inspiring talk. And I wonder, because all the examples you showed were from southern part of the world, but do these initiatives also work here in Europe? I mean, are they also developed, and do they work in Europe? Because I see it is wonderful, but I don't know here at home initiatives that replicate this theme, but perhaps it's because of my ignorance.
No, that's a very good question. I think it depends solution from solution. So biogas plants, that's very common in Europe, in Northern America as well. However, for example, in Europe, we don't have smaller scale communal type of biogas plants. It's something which me and my team are actually past 5 years working on quite intensively to create more modular systems for the European context. There are some different challenges in technical engineering challenges, but we are getting close to have first pilot up hopefully in next 2 years. So this is already, it's a smaller solution, but still pretty big solution. Then when I would think, for example, about a black soldier fly or different insect related aspects, that's very common in Europe, actually. I think Czech Republic and France are one of the biggest producers of the insect and especially then the protein afterwards. Here in Europe, there is not that big market of buying the live insect, but the protein from the insect, it's already quite a big market, and France, Czech Republic are among the biggest in that. And biochar, it's also one of the things which is relatively active around Europe. Interesting thing about biochar is that I would say it's coming in circles every maybe 30 years. So the circle has started, let's say, 5 years ago again, and more researchers and more companies are getting into the biochar now. There was not so much in past 30 years, but then before it was also a big topic. I don't know. I think it's very similar with fashion. Some things, they just come in cycles. So a lot of the things on different scale, they are also very common in Europe. Yeah. A very good question. Thank you.
Thank you very much, Professor Robic. Do you have more questions? Here we are.
Thank you for, I will say, this transfer knowledge. You talk about something we need, to fix this idea that the waste could be benefit. And in Germany, they import the municipal waste from Czech Republic, from Austria, from many countries, and pay money for this, and then they use it as a benefit. Actually, based on your research here and raising the awareness about using the right way to use this, somehow the resources, you say, not waste, what you recommend for using this byproduct from food industry, for example, and this the waste, which way it's better do you see, using the biochemical conversion or the thermal conversion or make combination? What do you think? So from your point of view.
Yeah, that's also very good question. And has many questions. It sounds simple, but it's complex to answer. So it usually depends on the resource itself. The thermochemical transformation we usually recommend more at the end of the process when nothing else really can be done. I think a couple of years ago we started also with the green extraction processes at our faculty. We have one of the 3 green extraction machineries which are in Europe, and we work with different materials to find what other compounds we can extract from them in an environmental economic way. And that's a good direction, because very often you can extract compounds for pharmaceutical, or food industry, or many others. It depends on the resource. It depends on the waste, let's say. But then it also depends where you are geographically. So, logistics plays a big role. So when you are talking about the waste which you have in some distant area, then it makes sense to transform it directly into energy, because everything else would be difficult in terms of logistics. So it's a complex question. Maybe that's also the reason why most of those topics has so much of research and so many publications you can do on that, because a lot of the questions you cannot just answer with a simple yes or no, or do this or do that. It depends on really a lot of factors. But a good question. Thank you.
Thank you very much, Professor Rubik.
Thank you very much.
More questions? Okay. If you haven't got more questions, then I will kindly ask Professor Ying Wang to present results of her research titled Control of the Oil Content of Fried Dough Sticks through Modulating Structure Change by Reconstituted Gluten Fractions. Professor Wang, please present your work.
Hello, everyone. I appreciate the opportunity to be here today. I'm a second-year doctoral student from Henan University of Technology, and my name is Wang Jing. The topic I want to share with you today is how to control of the oil content of fried dough sticks through modulating structure change by reconstituted gluten fractions. Well, first, let's take a brief look at the history of frying. The frying process dates back to around 1600 BCE, when it was practiced as a traditional manual craft. Nowadays, frying technology has become highly developed, and today's market is increasingly oriented toward health-conscious products. Therefore, we must pay close attention to 2 core issues facing the fried food industry: that's safety concern and the healthiness concern. According to the market research data, the global fried food market has demonstrated steady growth. increasingly from USD 28 billions in 2050 to USD 35 billions in 2020, reflecting a compound annual growth rate of 3%. The market is projected to reach USD 42 billions by 2025, indicating continued expansion in consumer demand and industry scale. In terms of regional distribution, North America, Europe, and Asia are the primary markets for fried food consumption worldwide. Among them, the North American market is dominated by the United States. The European market is led by countries such as Germany, France, and the United Kingdom, while the Asian market is primarily represented by China, India, and Indonesia. Next, we take a classical Chinese traditional food, fried dough sticks, 油条, as an example to introduce today's topic. The flowchart on the left illustrates the production process of 油条. Due to its relatively simple and industrially scalable preparation, 油条 has gradually stepped beyond China's borders and entered the international market. It can now even be found in fast food chains such as KFC and other global franchises. So, have you ever thought about how much oil a single fried dough stick absorbs, and how we can reduce that without changing its texture? In our previous study, we investigate the effect of varying wet gluten content on the oil content of fried dough sticks. We found that those with higher water absorption, longer development and stability times, and greater deformability exhibited denser glute network structures, which effectively inhibited oil penetration. Furthermore, principal compound analysis revealed a relatively strong correlation between the oil content of fried dough sticks and the proportions of gliadin and gluten in the protein fraction. To validate this hypothesis, we plan to conduct subsequent studies by adjusting the ratio of gliadin to gluten. Therefore, we adopted a separation and recombination experimental approach while maintaining the mass ratios of protein, starch, and water-soluble components in the raw flour unchanged. We varied the mass ratio of gliadin to gluten within the gluten-protein fraction. This allowed us to investigate the effect of different gliadin to gluten recombination ratios on the properties of the reconstituted dough and the oil content of fried dough sticks. The study found that when the ratio of gliadin to gluten is 5:5, the dough exhibits minimal mass loss and relatively good thermal stability. However, when the gliadin proportion exceeds that of gluten, the thermal stability of the gluten network decreases, indicating that the internal structure is more prone to damage during high temperature frying. This leads to the formation of larger pores within the fried dough sticks, which facilitates increased oil penetration. As indicated by the red arrows, these larger pores facilitate the rate of water oil displacement, resulting in increased moisture loss and greater oil intrusion. It was also observed that gluten networks with an extended gluten protein membrane structure, as highlighted by the red boxes, exhibit better resistance to oil penetration. Furthermore, under 800 magnification, the fried dough sticks at this ratio show the membranous substance adhering to the sample surface as marked by the green boxes. Laser confocal images provided a more intuitive visualization of the gluten network under different gluten to gluten ratios. When gluten predominates, the gluten network exhibits greater resistance, with the CZ2 gluten mixture significant thickens. The decrease in soluble large and some aggregate proteins observed in liquid chromatography results further suggest that morphology of the CZ2 gluten network may be caused by GMP depolymerization. Meanwhile, the CZ3 gluten network begins to stretch, and the CZ4 and CZ5 samples form more open gluten networks composed of finer gluten chains. The viscoelasticity of the dough depends on the gluten to gluten ratio. A higher content of bed sheet structures in gluten is one of the key factors maintaining good dough elasticity. CZ1 and CZ2 contain abundant bed sheet structures, whereas CZ4 and CZ5 are richer in alpha helix structures. Additionally, bed sheet structures formed at the expense of bed turns also contribute to the stability of the gluten network. Grip recovery tests further demonstrate that higher gluten content is unfavorable for maintaining the dough's elastic properties. Moreover, relatively higher amounts of soluble low molecular white gluten subunits are responsible for the higher storage modulus observed in dynamic rheology, which is also supported by electrophoretic bond patterns. Changes in the gluten to gluten ratio also lead to variations in the molecular interactions involved in protein network formation. When gluten exists, gluten ionic bands, hydrogen bands, and hydrophobic interactions and covalent bonds are all increased. Due to the openness of the gluten networks in CZ4 and CZ5, they require more hydrophobic interactions and covalent bonds to maintain network stability, whereas CZ2 requires fewer hydrophobic interactions. The internal environment of the dough and the content of GMP are both factors that influence the dough structure. As the gluten to gluten ratio changes, steric handles is altered with the gradual extension of the dough network structure. The exposure of hydrophobic groups increases, thereby promoting the adsorption of oil molecules by these hydrophobic groups. Therefore, according to the above work, we summarize two oil absorption mechanisms, which are water oil displacement behavior and the adsorption of hydrophobic groups to oil. A higher content of gluten promotes the displacement of water molecules by oil molecules, leading to a more open and extended gluten network. At the same time, this increases the exposure of hydrophobic groups, thereby enhancing the adsorption of oil molecules by these hydrophobic groups. Based on the conjecture and the problem we mentioned at the beginning, we have the following three conclusions. This research provided a foundation for developing specialized low-feed wet flour and improving the quality of fried dough products. Finally, I'd like to express my heartfelt gratitude to both my master's and doctoral research teams. I truly cherish these two big families. I especially want to thank Professors Wen Jiping, Zhou Xueling, and Liu Chong, all of whom are kind and charitable mentors. Additionally, I warmly welcome everyone to visit China and especially to Henan University of Technology. That's all for my presentation. Thanks for listening.
Thank you very much, colleague Ying Wang, for presentation of these very valuable results, because as we know, absorption of oil by fried food is very bad for our health. So do we have any question to colleague Wang? Okay, if you haven't got any question, then I will close this section and I will invite you to poster session and coffee break. Thank you very much for your attention.
Hello, everyone. So I think now you get ready for the last session of the conference today. And for the 4 sessions, we will have 3 presentations given by the speaker from Ghana and 2 speakers from Malaysia. So I just want to say a very short introduction about myself. My name is Vivu. I'm from Nông Lâm University from Vietnam. So it's my honor to be here to be the chairperson of this session. And I won't let you wait for too long. I would like to invite on stage Dr. John Lewis Zika Jaku and to give his presentation. And his presentation is about recent approaches for detecting complex food adulteration with machine learning and VIS. And I am maize, white, melon seed form, and calcium carbide. The stage is yours.
Thank you very much. So I'm Luis from Ghana. I bring you warm greetings from Ghana. I'll be making this presentation on behalf of my research team back in Ghana and my collaborators at the Department of Physics and Department of Measurement and Physics, Process Control. Before I start, I would like to just get all of us to pause for some few seconds and imagine that perhaps the juice that you bought yesterday, which had a very bold label on it that natural, sugar-free, and green, was actually just maybe some liquid with water and extract. You know, it's tricky. Yeah, or perhaps maybe the bread that you ate this morning contained potassium bromate, which can give you cancer. Or even maybe the fact that the coffee, you've had about 6 cups of coffee this afternoon, but you're still tired and sleepy because there was probably some fly in it. I mean, these are just wild allegations, but sometimes these adulteration practices still happen. Today, I'll be talking about adulteration of honey, banana, coconut, melon seed powder, and then wine. So food adulteration is basically trying to find out whether you're getting the value of what you are buying, okay? So it can be in the form of dilution, addition, replacement, subtraction, you name it. But according to the definition of Codex, the food security, to achieve food security, they must be affordable, safe, and nutritious products. Okay, so I'm going to focus on the safe products. So there's always been a constant force field of analysis between producers and the regulatory authorities. Okay, so some fraudulent producers are always trying to put consumers at risk by selling fake products or products that are not real or inferior. They sell it at a higher price, or they use inferior products and sell it at an equally higher price to you for profit. So when you take it, you don't get that organoleptic feeling. So you are being denied of the sensory benefits and the nutritional benefits. On the other hand, the regulatory bodies are also working hard to earn consumer trust. And because of that, we want to guarantee that consumers are safe, and also we can reduce food waste. Because what happens when you buy a product and you taste it and realize that it is fake, you discard it. And it all contributes to waste. So food adulteration is actually it's a global problem. Okay, it's you can call it a behavioral cancer because it continues to grow, and you can see from research over the years, it has gained a lot of attention. It has gained a lot of attention. Researchers are constantly trying to develop new ways to detect these complex food adulterations. Because existing methods such as these are quite expensive, they require high technical expertise. They are also, they need reagents, maintenance, and all that, so it's expensive, and some of them have sensitivity issues. So there is a paradigm shift. A paradigm shift means a gradual interest from one area towards another area. And you can see this researcher is very happy because he no longer needs reagents, which is going to cost him less. So compared to the conventional methods, emerging techniques such as sensor-based devices, near-infrared spectroscopy, we had a few talks about it today, even 3D printing. So these are some advanced techniques. Also, they are more precise. The advanced methods are more precise compared to the conventional methods. One of the best advantages is that they can be non-invasive. So you don't necessarily need to go destroy the product, do some extraction. No, you just put it on it and then you get your reading. Just like the sensor device that was measuring apples this morning. Above it all, we have the machine learning advantage, which can incorporate cloud systems so you can have your analysis done remotely. So there are a lot of these advanced techniques, but I will focus today on spectroscopy, near-infrared spectroscopy. That's basically having a measurement within electromagnetic spectrum in the near-infrared region. That is from 700 to 2,500 nanometers, because they are important absorption bonds that can fingerprint or give you an exact idea about the chemical or physical or biological composition of the product that you're analyzing. So I'm going to talk about application of near-infrared spectroscopy to detect adulteration of 5 types of different foods, starting from wine. The QR code means that it's a published paper, so you can just scan it and follow on the original paper whilst I talk about it. So this was a study done to detect aflatoxin in white maize. Sorry. Maize is it's a staple. It's one of the most common food products or the most common grains you can find across the world, but it's also vulnerable to fungal infection, which most of the time, not always, but most of the time can lead to mycotoxin contaminations. Yes, for example, aflatoxin, which is hepatotoxic, that means it can damage the liver. Now, mostly these mycotoxins are determined with conventional methods, which we talked about before, such as HPLC, but we already highlighted their disadvantages. So we are trying to apply NIR to detect this. So basically, it was a quasi-experimental method. That means that we designed it, had control of variables in the lab. So we got a yellow maize flour and white maize flour. Then we did a spiking with aflatoxin through a serial dilution. Then we measured it with the NIR. So we had different adulterated samples within these concentrations because we wanted to cover the upper and the lower boundaries of the EU limits for this toxin. So from the analysis, we could see that even from the raw spectra, we could see some important peaks and absorption bands that could be related to some of the biochemical components of the food. Some of these important bands were the wavelength as 1,150, 1,950, 2,100, and 2,350. So we went ahead to do a linear discriminant analysis, which is a classification method, supervised classification method, to try to see if we could classify or basically detect these different concentrations of aflatoxin in the maize. And we realized that we could do it with a high prediction accuracy of 91% after cross-validation, also with sensitivity, acceptable sensitivity levels. So basically, what we are trying to say about maize is that it is possible for you to detect aflatoxin contamination in maize. So what next? So we advise probably that we need to create region-specific spectral libraries where we can have like a cloud system where anybody can generate their spectra in there. We can also deploy software for real-time aflatoxin analysis. And lastly, to pursue regulatory authorities to convince them after further studies that indeed this is a pursuable path. Next, I will talk about predicting ochratoxin in white wine. So similarly, these wines, produced from the berries. Sometimes the berries can be infected with fungi. Some of them are good, like the brutized wines, but some of them are also harmful. So this time we focus on the harmful one, ochratoxin. So ochratoxin is also, it has been reported to end up in wine sometimes because it is stable and it can sometimes withstand the processing. So it's not a surprise that it's becoming a problem. And it's also a human carcinogen. So basically, we got white wine, spiked it similarly with ochratoxin this time. These concentrations again to cover the EU limit. And this time we went ahead to use a portable handheld spectrophotometer to scan it. So we did the analysis at 3 different wavelength ranges. Basically, we wanted to test the possibility of detecting it at the Vicinal range, and at this range where they sometimes refer to as the aquaphotomics range, and then also at the main NIR range. Within the wavelength range 1,300 to 1,600, and also within this wavelength range, we had better classification accuracy, suggesting that VCNIR may not probably be the best for this sort of measurement, but probably NIR itself. Yes. And also we realized that the NIR spectra reviews give some information about the physicochemical components of the white wine. So we also encouraged similarly that specific spectral databases be built along with real-time analysis using handheld devices. Also, we did some study on calcium carbide. Calcium carbide is banned. It's a dangerous chemical because it causes cancer. It's mostly used in the filtering shops, but unfortunately, in some parts of the world, some people use it to ripen fruits, not just banana, but we applied it on fruits. So basically, when you put the fruit next to the calcium carbide, it increases the release of the ethylene and it causes it to turn yellow, but it's not actually ripe. And sometimes it is harmful. So we decided to do a study on this. And unfortunately, we realized that there was some regulatory gap because there was really no standardized method to determine calcium carbide in fruit. So this was the first of its kind. And in Ghana, we did a survey in Ghana and we realized that there were mainly 2 types. So basically, some of the commercial vendors said they wrap the fruits with the powder. Some of them said they soak it. So we tried these 2 techniques. So we mimicked this in the lab, went ahead. So we did that with 2 different varieties of banana. We got a foreign variety and the most common local variety. We did at a natural ripening. That means we left it to ripen naturally. Then we kept some of the banana in the dark because it's also a common practice to keep it in dark rooms. Then we tested the carbide ripening as well. So we did some physicochemical analysis. Basically, this was the flowchart of the study. So we realized from physicochemical analysis that the brightness, color L, tend to be the highest when it was soaked. So or wrapped. So with carbide treatment, we saw that there was an increase in brightness, almost the same as the natural ones. That means that physically it would be almost impossible to tell whether this is naturally ripened banana or it is artificially ripened illegally. Yes, from our classification, we could also distinguish that this banana was ripened in the dark room, and that was ripened naturally, or that was ripened with carbide. So this was a good step for us. We went, so from our classification accuracies, we could see that the accuracies were all above 80, which means that the model was quite robust. And we also did the analysis with, so we scanned the banana with the peels on and also without the peels. And either way, we still have these accuracies. So we went on to predict the color with partial least square regression. And then we got R squares greater than 7 with low errors and RBPs greater than 1.9. So basically, we are trying to say that it is actually the first step, because we haven't really seen, before this, we didn't really see any study on carbide with NIR. So we think it's an important step and probably requires some further studies with other fruits, especially green fruits like orange and other stuff, so that we can see whether maybe this trend is something to really worry about. Yes, we also have the melon seed, which basically is it's a delicacy in Ghana. Because it's highly proteinous and it's used to prepare a lot of different varieties of fruits. But unfortunately, there has been reported, there have been some reported studies of adulteration. People combine other flours and burn products in it. So we also got the pure melon seed, milled it, and we tried to adulterate it with what we call the soybean powder, and then the gari, which is cassava, milled and dried cassava. For those of you who do not know cassava, this is how it looks like. It's a root and tuber crop. It's very rich in carbohydrates. Yes. So also from the spectra, we could see some important peaks, which gave us some ideas about the physicochemical analysis even before we went on to do further analysis. And from the classification, we could see that all the tested different concentrations of adulterant could be classified with good accuracy. So it goes ahead to tell us that NIR indeed has good potential as a forensic tool. Lastly, I will talk about honey. This is a bit surprising because we came across this in some literature that in some parts of India, they adulterate it with foam. This is very common in Ghana as well. So we decided to do a survey, because in Ghana, mostly when people wish to discard their mattresses, it's just they can just dump it right in front of their homes, and then some producers go and pick them up, and then they burn it. So when you burn this one into the honey, it mixes so easily you can't even see the difference. So we decided to try this with this NIR. So we went to we got an authentic honey, came up with some different concentrations, which we designed, measured it in the lab. Then we did a PCA. So from PCA, everything looks overlapped. And then we went ahead to do an LDA. And then we realized that pure honey, which was is black somewhere in the middle here, seemed to overlap with 1% and 1.5% honey containing mattress, which means that if small concentrations of this foam is used, even NIR will struggle to detect it. And note that NIR is an advanced technique. Also, we went ahead to realize that the samples containing 3% and 5% were close to this market sample. Although it didn't overlap, it was close. So we are still trying to dip further and see whether actually it contains foam or there's just something else. There's also a market sample away somewhere here, which was completely different from all the other market samples, even though it's supposed to be the same honey variety. So we have no idea what is happening there. We don't know whether there's something else in it. So we are also still working on that. So we went ahead to do a partial least square regression, and we could predict this foam in honey with good accuracies, with R squares greater than 0.9. So basically, what we are trying to say is that NIR is the next generation forensic tool for food surveillance. And we need databases or region-specific spectral databases that can be shared for mutual benefit. Imagine walking to the supermarket just with your smartphone, and you can just scan this coffee, and it's written Arabic, but when you scan, it tells you it's Robusta. Yeah, I mean, that's next generation digitalization. So I would like to end here, but I can't end without acknowledging my university for the support. Also, I'd like to acknowledge colleagues and friends at MATE, the Department of Measurement and Process Control, especially Professor Zoltan Kovac, who is like a model to me, he's my mentor and a brother as well. I also like to acknowledge BioSys for the opportunity and the invitation to be here. And then Erasmus also for covering some of my trip costs. And lastly, my tiny research team back in Ghana. These are my postgrad students. Yes, so thank you very much. And if you have any questions, I'll be happy to answer. Yes.
Thank you so much. Thank you so much for very nice presentations. I would like to ask the audience, do you have any question for this speaker today? So I had a small question for you.
Thank you.
Because I'm saw that you had experiment with the compound you want to detect, but in the artificial way. So in case if the product is like the blendings of a different compound, and what do you think if the research from NIR can be influenced, the accuracy?
I'm sorry, but can I get a question again, please?
Oh, yeah, sorry again. So for your experiment, you do with the artificial way.
Yes,
That you put the compound to the fruits, and then
That's
you
correct.
detect by NIR.
That is
But
correct.
in case there are some product like already has the different compound,
Yes.
and what do you think if the other compound can influence the accuracy of NIR methods?
Yes, thank you very much. So I get what you're saying. So NIL can be both targeted and non-targeted. So in most of the instances in our research, we focus on the targeted. But you could, if you, for my last presentation on the honey, I mentioned that there was a market sample that was completely different from the others. So even though it was non-targeted, even though it was targeted, we got a non-targeted result because that market market sample we noticed was completely different, but we don't know what it is. So then we would dig deeper. So of course, if your target compound is not there, you will still notice it. So it basically gives you a fingerprint. So anything that deviates from the original, you know that it is not appropriate. It's not authentic. I don't know if that answers the question.
Yeah.
Get
it. So
Cool.
thank you.
All right, sure.
Okay, thank you so much for your presentation. So thank you again. And I would like to invite our second speaker for session 4, Dr. Waffle Duming, to deliver his speech. Welcome on stage.
Okay, good afternoon, everyone. Again, my name is Walter Dumin, and I'm from Malaysia. And I guess my talk today is more on sharing because our work is more specific to some region part in Malaysia and in Sarawak. So I hope that dear colleague can learn something from our experience as it is more specific in the region. All right. My topic today is more on primary screening and characterizations of a disease that a causal disease that affecting a pineapple plant in northwestern regions of Sarawak. So this is the overview. We're gonna go a very brief on this overview, introductions and disease problem in Sarawak, especially primary screening, a methodology that we conducted, as well as a result and together with the discussion. So if you look over here, for those who, just a reminder, this is the pineapple look like that we're working on. And then if you look on this side, it's the benefits of the eating pineapple. And then just for introductions, this is the country that mostly produce pineapple product. And then if you look over this side, Indonesia, Philippines, and Costa Rica among the main producer for this. Now, in Malaysia, we have this one body that we overlook oversees the pineapple industry that we call Malaysian's Pineapple Industry Board, which is look after the development of this industry. And you look at it, oh, sorry. If you look on this slide, the Malaysia has divided into two parts. One is a Borneo, and then another one is a Peninsula. And the biggest producer for Malaysians is a place we call it Johor, a state of Johor. And another one is a second one is we call it Sarawak, where we our locations and our research area has been conducted. So just for information, this is the variety that's the most popular in Malaysia. And for now, this is the variety that it's mostly cultivated, which is we call it Morris and MD2. And this is a choice because it's sweet, juicy, and it's small, which is fit to the consumptions or the taste of the Malaysian people. So in Sarawak itself, if you look at this is the area, and then this is the a brief statistic that the district that produce pineapple in Malaysia. And if you look over here, this is the most cultivated. And you look at this one, one of the variety is embedded the name itself, which is indicate that this is the one of the most cultivated variety in Sarawak, together with the MD2 and Josephine, Morris, as well as the Sarawak over there. All right. So why do we focus on the pineapple? So recently, especially after the COVID-19, we realized that it's important to focus on the food security. So the Sarawak government, state government, now decided to focus on 4 crops that need to be focused on. So one of them is the pineapple. So that's why we looking into it now, and then we try to develop it in a way that it's gonna what do you call it? Fulfill the demands in Malaysia or even worldwide. So disease problem in Sarawak, this is the disease that has been reported so far. And then you can see all of these things, all of this plant has been infected by this particular disease, Phytophthora, fruitlet coral, black root, and so on. So if you notice over here, I put it in this on the upstairs here. This work has been conducted by a researcher from Sarawak Agriculture Department in 2009. So it's been like 16 years already. So because of the length that has not been done anything, we believe that there's a research gap that need to be updated on this one. So so we hypothesize that since this has been 15 years, there's nothing what do you call it, a formal report, because if you go to the Department of Agriculture of Sarawak website, they still put this one as their formal report. So we hypothesize that it's actually there are already new diseases in Sarawak, but it's not been identified or it's been probably overlooked. So in this project, we put our main objective to identify and characterize the potentials of new pathogen responsible for the pineapple disease through morphological and molecular identification techniques. Since the molecular identifications now, it's much more affordable, it's faster, and you don't need a high expertise to identify them. So we more focus on these techniques instead of the morphology. So this is the methods that we working on. We collected a sample from different regions in Sarawak. I will show which region later on. And then we isolate them. We obtain the pure culture through the technique that has been listed above below. And then we, using the morphological and the molecular identifications, we narrow down what is the what isolate that we are looking on. So for morphology, for the fungal, we use internal transcript space as a molecular marker to be targeted, whereas for the bacteria, we use 16 sRNA as a molecular marker as a target. So often obtain the sequence, we use a blast and phylogenetic tree to identify. And in order to complete the Koch postulate, which is confirm the pathogenicity, we conducted a varietal test for that. So like I said just now, this is the Borneo Islands, and this is part of the sample that we took. We took a sample, and this is the area where a 4 area that we have collected, which is indicate that this area is actively involved in planting or cultivating pineapple. So in the result, we have collected, obtained more than 100 pure isolate, but we not be able to sequence all of them due to the limited resource. So we selected approximately 65 of them to be sequenced, and then fungals 26 isolate and bacteria 39. And then for fungal isolate, 80% of them is not pathogenic. 20% of them they have a potential group or family of fungus that can cause like Diosporidia, Fusarium, and Penicillium, and this. So for bacteria isolate, we sequenced 39 of them. And then if you look over here, 42% of them is non-pathogenic, but it's more than half of it, it's pathogenic. So this is the area the isolate that we have been identified them. And then if you look at this, most all of the area that we have isolated indicate the presence of this. So the interesting thing is all of these 20 isolate is belong to Dickia, which is known to be a very devastating disease in pineapple worldwide. So because of that, it's draw our interest to further identify this, since the rest is not that common that we found. So just for information, Fusarium and Penicillium is already identified. To be honest, this one is never been reported in pineapple, but we found that this family can cause disease in the tropical fruit. So we put it as opportunistic new fungal pathogen, which is might be probably can cause in the future. So this one, the Diosporidia, this has been recently identified in 2002. It can cause a leaf blight in pineapple. That's been reported is in India. So we categorize them as a new candidate for the fungal pathogen. So for the Dickia family, we further identify them. We want to confirm that it's a Dickia family. So we further conducted using a PCR using the ADE and ADE2 primer, which is developed by NASA in 1996, which is focusing on identifying the family of this species. So this is an example of the isolate that we have obtained. And then we look over here, it's all positive of them. So we sequence the 16S and we constructed the phylogenetic tree. And you look over here, 3 of them. We only pick 3 because it's 100% similar. So we don't put all of them. It's considered same bacteria. So if you look over there, it's belong to Dickia Z family. So it's confirmed the family. But then how about the pathogenicity? So the pathogenicity, we look over here, we try into 1, 2, 3, 4, 5 variety. We name them based on the local name. It is quite weird because we have a name Thailand over there, but I'm not sure why the local use the name Thailand. Probably it's been taken from Thailand. So based on this result, all the Dickia that we have isolated confirm its pathogenicity. So now, why it is important in Sarawak? The most important thing is because this finding is to try to avoid, not try, it is avoid the misdiagnosis of the disease, because the symptom for Phytophthora and the Dickia is about the same. So most of the time, because the Dickia has not been identified yet, so what happened is people always thought that it's a Phytophthora, so the misunderstanding caused them apply a wrong countermeasure. So another one more is that it is a crucial information for the disease management, because until now there is no bactericide or chemical treatment that has been approved by the government to treat this problem. So because of that, if it's been detected in the field, we normally advise the farmers to focus on integrated culture practice ataupun sanitation practice. Okay, so this is the only that we can do since there's no chemical treatment available currently. So it's also provide us more confidence to the industry for smallholder and private investor, because once we identify that, they know what to do, and the management strategy will be more thorough to control. So another one more is that we can target the phytosanitary measure that is provide a more interest or it's provide more confidence to the international standard. So just a photo of the area that we have collected our sample. And since I'm here, and I would like to introduce our university with a few slides. This is our university, which is University Malaysia Sarawak, with the acronym of UNIMAS. And we have a 12 faculty, which niche of biodiversity, sustainable community, and information communications, and creative technology. So this is our location over here. This is the heart of Borneo. This Borneo divided into 3 countries, Malaysia and Brunei over here, and over this side is Indonesia or Kalimantan. And so this is where the faculty where I come from. We have a 6 undergrad program, 2 postgraduate program by research, and postgraduate as a coursework. Although recently we have added a few more. So another one more, which is appreciation for me. It's UNIMAS Agro Research Center that they've been recently established. So we included all this topic niche area that we can work on. Then I hope that if any interested, we can do more collaborations on that. So that's all for my presentation today. Thank you. Or we call it terima kasih in Bahasa Malaysia. So thank you.
Terima kasih, Dr. Hua for doing. So I would like to ask our audience today. So do you have any question for our speaker? So for me, also small questions. I'm curious because you mentioned in your abstract that the cultural agent is come from India.
Oh no.
No.
It's actually one of, if you Legispodia, it's one of the fungus that we have identified, which is indicate that it's pathogenic to pineapple is India. That's been reported in India. So we assume, or probably we expect that there might be, when we found the similar strain in Sarawak, there might be cause a disease as well. So we looking on that, we will try to test that, because we focus on the Dickia first. The presence is very significant in Sarawak, and need more attentions currently. So that's why we stress that it's supposed to be.
Good,
All right.
thank you. Thank you so much.
Thank you.
Thank you. So it's come for our last presentation today. I would like to invite to stay Abu Hanan bin Zulkarnain to deliver his speech.
Okay, how are you, everyone? Tired? Okay. Okay, so thank you, Chairperson. Thank you, the organizers, for giving me the opportunity to give a speech for today. Today, this is a part of a big research, but I want to focus only on the comparing between the eye tracking and the virtual reality eye tracking on sustainable labels. So today, this is my presentation roadmap. Mostly, I will be talking about what is the sustainable labeling, and then what is eye tracking, screen, and also versus VR methods, and then the study of design and methods, and also the key findings, practical implication, and also the conclusion. So first, this is a real case study. I want you to think, okay, we have two types of brand, the Tropicana brand, if you know about this, because this is most famous in America. So in 2019, this Tropicana is redesigned their packaging, and within 2 months, they lost around almost 30 million US dollars. Do you know why it happened? Because of the old brand, you can see the logo and the brand itself, you can see it's on like a center of the attraction. While the new packaging, the brand is at the side. So people will like thought, okay, this is not a Tropicana brand. This is some sort of a different kind of a knockout Tropicana brand. That is why this failure that is have been the Tropicana to redesign back to the old branding back. So because it give back their profit. So this is why I want to talk about sustainable labels. So sustainable labels is like a visual certification of a food packaging that is that the product is being certified that okay, this using environmental or socially responsible practices. So there's a much more sustainable labeling. This is just an example. So so the aim of this study is to compare between the normal desktop eye tracking and also the virtual reality eye tracking. So as a question, do sustainable labels receive more attention in the VR or on the screen-based eye tracking? And then does VR lead to better recall or higher willingness to pay? And also the last question, the research question is, are label effective and the visibility consistent across the mediums, either the VR eye tracking or the normal eye tracking? So this is eye tracking. This is a simple basic eye tracking that I can present to you. So eye tracking is where you record your eye movement to see how long people look at certain things. So for example, this can reveal subconscious of attention. First, we have the first fixation, means where you see the item first, or the dwell time, which you spend the most time on the certain parts. So this is the subconscious of the eye tracking. So usually in the food research, eye tracking is being used in the nutritional panels, brand or logos, and also the sustainable label as well. So there is three types of eye tracker. We have the screen-based eye tracker. This is where the eye tracker that you put on the screen of the computer, and then it measures your eye movement. And then we have the wearable eye tracker, which you use a normal like a glass, and then you can walk around with it. And lastly, we have the virtual reality integrated with the eye tracker as well. So next is about immersive technology. So this is a reality virtually continuum by Milgram and Kishino in 1994, which the reality is where we are now. We are in the reality. So next is the augmented reality, where you can see all the reality, but there is some digital elements. For example, like the Meta Quest Pro or the Apple Vision. This is the AR or augmented reality. Next, we have augmented virtuality, which is inverse, which the what's it called, the context is changed, but you can still see the real product in front of you. This is augmented virtual reality. And lastly is the virtual reality, where all is the digital items. So you are immersed in the some situation. So we are focusing on the virtual reality at the moment. So this is the screen-based and also the VR eye tracking. So for example, the screen-based eye tracker, you can see 2D image on a monitor. Means you are in the laptop on the monitor, you see the 2D image, and then the participant need to sit still, and they need to have a control lighting and also the distance between the eye tracker and yourself. So where the VR eye tracking is a 3D virtual environment. I'm not sure you can see, but there's some of the round circle that is detect your eye movement. And then usually using the HMD or the head-mounted display or the VR headset, it can be free moving, and also you can move freely. Means you can just move around, walk around, and also you can look at up and down. So this is the advantage of VR eye tracking. And also the last thing is the ecological validity, because you are immersed in a almost normal situation that you are, for example, like you are in the living room, you put a living room environment, so you are more adapt to the environment that you are immersed in. So this is what is the VR eye tracking can do. Next, we will do a preliminary studies, which we ask a normal basic question, which sustainable labeling, food sustainable labeling that they are more familiar with. So as we see, we what's called ask 85 respondents, and we choose based on the most that is familiar, the moderately familiar, and the least familiar. So that is why we have 6 labels that we are choosing to do for this research. So for this research, we are focusing on the 3 products because, for example, 3 known products, for example, like the Cadbury, this is, we change the sustainable labeling there. So it can give like a participant to see where they see the sustainable labeling from. So for example, like the EU organic, this is the EU organic. This is the most familiar for from the respondents, where the Rainforest Alliance is, this is the most familiar. So we choose the product based on the suitability of the product itself with the logo. So for example, Rainforest Alliance is most suitable with the coffee product, for example. So this is why we edited based on these 2 products. So the eye tracking that we use is the Tobii Pro Nano. This is a normal Tobii company. And also for the VR, we use the Pico Neo 3 Pro Eye. So where this is the VR integrated eye tracker. So the metric that is captured is the fixation count on label and also the total fixation duration on the label area. So we want just to focus on the sustainable logo itself. So as the methodology, we recruited 42 participants to do sensory, not sensory testing, but to do visual tasting, which we have 20 different random products, which we integrated the 6 product of the sustainable labeling. So this is to prevent bias, so to prevent that they recall this product. So that is why we combine it with the 20 different kind of products. So we repeated twice. One is a normal eye tracker and one is in the VR. So this is a normal eye tracker that you can see, and this in the VR eye tracking. So as a result, as you can see, the most highest people that see most count that we can say that is they are more towards the logo is the non-GMO, and the least is the leaf. So what am I meaning about it? This is based on the heat maps, for example. This is a screen-based eye tracker, which you can see the most fixation count here in the screen base compared to virtual reality, because the logo itself is on top. So which in the virtual reality, the logo, most of the people are looking on the brand itself. So as you can see here, also the red color is where the people look the most, and the green color is where people look the least. So this is why, as you can see, for the non-GMO is the highest on the logo itself, while the fair trade is more dispersed in the virtual reality eye tracking compared to the screen-based, because in the screen-based is more focused. And then this is the Euro leaf. Sorry, this is the Euro leaf. Yep. And yes, they are more focusing on the brand itself also compared to the virtual reality eye tracking. Where this is a UTC certified. Also, they are more focusing on the written because the logo is here. A Bose product, they didn't look at the logo that much. And this is the rainforest, which, yes, for the eye tracking, they look the most, but compared to the virtual reality eye tracking, they didn't look a lot. And lastly, this is the leaf, which they are more focused on the wording and also more focused on the coffee itself compared to the eye tracking. Also, they didn't look at the logo itself. So we did some statistical analysis. As you can see, when we are comparing between the logo itself, there is significant differences. And when we compare between the condition, the eye tracking and also the VR eye tracking, there is also a significant difference where they are more prefer the eye tracking, not the normal, the screen-based eye tracking. But when we are doing the integration between the logo and the condition, there is no significant difference between the normal eye tracking and also the virtual reality eye tracking. So this is the good news for us, because no matter what you do, either with the normal eye tracking or the VR eye tracking, there is no significant difference. So, and then we go to the PCA. As you can see, for the first factors, these capture the most on the visual attention compared to the second factor. This is more distinguish between the screen-based and also the VR-based. And yes, for the practical implication, this is from the comments, actually, from the participant, that saying that for the eco label, they suggest to do more bold colors or more closer to the brand itself, because when you see the big brands and you put the sustainable logo beside it, so people can see on the big brand itself compare when you put further from the brands. So, and also when use VR as a pre-marketing testing. So for example, like you don't need to print out, because when you are in a VR, you can design or stimulate the environment, which you can just focus on the model itself, compare when you need to pay for printing, for example, and all those like more cost on it. So that is why virtual reality is more efficient. And also apply the evidence-based validation to use the eye tracking to confirm the design, compare to just assume and you print it, and then when at the end it will fail. Compare, for example, like the Tropicana brand. And as a conclusion, desktop eye tracking is more precise because it's more focused, but the VR eye tracking is more dispersed because you can move freely, you can look around. So this is why if either you use both of the eye tracking system, it can be beneficial, but it being preferred to use the eye tracking, virtual reality eye tracking. So as for future research, we want to investigate more on the consumer attention to the ultra-processed food, for example, and the Nutri-Score labels in the VR's eye tracking, and as well as because nowadays there is also digital labeling is an upcoming trend. So this can be also one of the future research for this. So thank you so much. If you have any question, feel free to ask.
Yeah, very interesting the presentation. Thank you. And so, do you have any question for Abdul? So I had a question for you.
Okay.
Do you observe any differences among the international participants who has different points of view, like the texture, the color, or the shape,
Yeah.
when you do experiment?
You mean by with the participant itself?
Yes.
Yeah, at the moment, we didn't apply on that because we want to see the technical part of it to compare between the eye tracking and also the VR eye tracking to see how most what's it called, like how they see this labeling itself. So yes, sometimes some participants said that they didn't see of sustainable labeling because it was too merged into the packaging itself. Because for the coffee brand, for example, it's a white color. So sometimes it's a bit like unnoticed. For example, compared to Cadbury, which have a more bold color. That's why they are more focused on that. So at the moment, that is our next step. But at the moment, we are only focusing on the comparison between those two. Yeah. Thank you.
Yes, and I had a notice from the organizer that who come here with car, you had the free ticket at the registration table, so feel free to take it. And so now I'm finished my task, so it's time for the others program. We will come to the next program's conference quiz, so try your best to take the prize.
Good evening, everybody. My name is Tamás Zsom. I'm responsible for. Okay. 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 I'm responsible for the conference quiz, so I was given the task for give you some fun. Okay, after all of these nice scientific researchers and nice presentations. So first of all, I would like to remind you for a very important task of you. So I do hope that you have already voted for the best oral presentation. Okay, if you're not, then please go for and first of all grab your phone and connect to the Wi-Fi system. And the password and the Wi-Fi name is given on the lower part of the presentation here. So BIOSYS, the Wi-Fi name is BIOSYS, and the password is for it's for food 2025. Okay, so first of all, just join to this possibility to the open Wi-Fi system, and then we are going to for a Kahoot conference quiz. But before that, please take your time, for example, for 2 or 3 minutes maximum, and vote for the best oral presentation. Okay, so this time is given for us and for you to vote, and for the scientific committee to summarize all of your votes. And finally, based on your vote, the best oral presentation award will be given. So if you are not familiar, what will be the prize? On your right, there is a table with 3 awards given from our sponsor, the Samos sweet Hungarian company. So for the best oral presentation, for the best poster presentation, and for the conference quiz winner, they are going to be given, a small present. Okay, so take your time and go for the voting if you have not done already. Please raise your hands. Who is done with the voting? Okay, who is not? Please go for that link which is given in your leaflet. Okay. Okay, but first of all, you have to vote, and then we are going to go for the play. Okay. Okay. So everybody is ready with the voting. Who is on the voting process now? Nobody. Okay. So we can go for a small play, a small fun. Okay. So let's check. Okay. visit, you know, kahoot.it, okay, for your play, and I will give you shortly, you know, the pin, you know, to log in and to go in. Okay, so as check here. Hold on, login. Ja, de már be is léptünk. Okay. Okay, so first of all, in library, this will be the one. Okay, so we are going to load the code. Okay. And okay, hold on, start. Okay, you are going to be given a PIN code, you know, to log into on the site of kahoot.it. And after you have logged in, you are going to be asked, you know, for personalize your attendance. Okay, so you can get some faces and hats and characterize and your account or something. And after we have, I do not know how much we are here, about 20 or 50 people. We are going to start with the questions, okay? You do not have to be afraid. Only basic elementary mathematics and intuition and picture recognition you have to use for your answers, okay? Okay, 42 people are joined. Okay, so hopefully, who is on the way to join to the quiz? Everybody is in the system? No, you are not going to play anything. Okay. Who is not playing is not going to get any reward, okay, or any rewards. Okay. Okay, yes, unless he was the best, or she was the best presenter, okay? So 44 people already joined the system, 45. Is somebody on with the logging in? Okay, so everybody is eager to play, okay? So let's start with the fun. Hopefully it will work. Okay. So only 2 seconds, and you are going to be given the first question. So if you saw three corners of three corners of a table, how many corners will it result? And you've got only 20 seconds, you know, to answer and choose the correct answer, or just take one. So if you saw three corners of a table, how many corners will it result? Okay, 12 correct answers you were given to us. And sorry, only 12. And 12 was the correct correct answer. And who is the first one? Julia Keresztúri is the first one now, but we have several questions on the way, okay? So, what number does the question mark replace? This is the second quiz. Mathematics, basic one, okay? Fast calculation and give your answers, please. The harder the question, the more time you are going to give in to do your answers, okay?
Okay. Some of you is still calculating, but the time is running up now. Okay, only 19 correct answers were given. So I'm going to give you a short solution. What was the trick in this mathematical calculation? Okay, so Vilmos is the first one now, and let's see what was the banana peel. Okay, so first of all, the first line is correct. The first line is easy peasy. So 20 values means a plum. In case of the second one, or in the third one, you are going to get for the bananas 9, and for the cherries only number 2. And in case of the last question or the sub question, there was the banana peel because there was a multiplication sign, not a addition sign. Okay, so if you just gave 51 or something, not the correct answer, it was the banana peel buried down there. Okay, so the correct answer was 58. Which one fits in the pattern? Okay, you just use your imagination and reading capabilities. Okay, so what will be the correct answer? Hurry, book, panda, or ruler? What do you think? Oh, the penny has dropped. Okay, almost 10 seconds left, to give you a decision. 3, 2, 1, and 27 correct answers were given. So it was a hard question, to solve. So let's see who is now on the first place. Okay, Julia is always on the top, so keep on fighting for the first place. Okay, so let's see. So what was the solution? The first one for E, and the leaves and the ends means elephant. Okay, the second one, or elephant. Okay, the second one, cat, ear, and a pill, meaning caterpillar. Okay, or something. Okay, and the last one, based upon the first two, what do you think? Yes, the orangutan is the correct answer. So orange and U and 10, orangutan or something like that. Okay, so it's a tricky one. Okay, so how much was it? 27 or something. The correct answers were given. So what was not on the table in the picture you saw earlier? Do you remember on the first picture? You just gave a tip. Okay, 1. Okay, only 12 candidates were correct, and the sugar bowl was on the table. Okay, sugar bowl was not on the table. Okay, pumpkin, blanket, and the teacup in the tea in the teacup was already on the table, but we didn't have any sugar bowls. Okay, so who is now the first one? Huey is the first one, but we have still questions, to answer. Okay, so keep on trying. So the next one, as I told you, what was not on the table? It was just, the sugar bowl. because we haven't seen any sugar balls on the table in this picture. So let's go on to the next question. What does the question mark replace? Again, simple mathematics, okay? Or just picture recognition, okay? So only 10 seconds left for your answer to give. As I told you, this is basic elementary mathematics or basic elementary. Okay, so much better results, 29. So what is the correct answer, or what was the correct answer? And who is now on the top? Okay, again changing. So you do not know who is going to be the winner. Okay. So why on earth was the correct answer given? So, you know, one cross, hair cross, or I do not know how to say it. You know, if you are a sniper, you are going to know what is it, or a plus, it means 1. So in case of the first option, you've got 9. And in the case of the last one, how much you have? It was 4, okay? So what does the question mark replace? Again, not a silly question. Again, you have to do some mathematics, okay? As I told you, basic elementary mathematics you do need to answer the question. Okay, the time is over. So only 12 questions. Only 12 candidates answered the question correctly. So why on earth was, you know, the correct answers number so low? But first of all, Balázs is the first one now. So let's see what we have here. So what is the correct answer? What was the correct answer? So if you were calculating in your head, you just have to skip it, because you just have to recognize, you know, the similarity between, you know, the two last options. So 1 melon and 1 lemon equals in both options. So 21 was the correct answer again in this case. So tricky question. First of all, you just have to recognize the problem, and then you should give, you know, the next answer. So we have only 4 questions left. Which area is the largest? Just summarized rapidly, you know, the numbers, or just try to give the correct answer. Okay. Only 1. So it was just a tip. Okay. So four different colors, 4 different shapes. So let's see who is on the first point. Okay, Balázs is always, you know, the first one. Okay, so let's see. Only small differences were given to you, you know, to recognize. So surprisingly, the red area was the biggest one. Okay, so first impression for me was, you know, the yellow one, because it's the most rectangular, but surprisingly, you know, the red ones were the biggest in area. So let's see the next one. So what is in the picture? Picture recognition. Seconds left. Okay, so did you recognize it? Okay, it was an easy peasy question. So 36 correct answers were given. So Yuli is on the top again. So let's see what was in the picture. Kiwi. Okay. So what is the solution for this question? Again, a tricky one. Okay. But you are given almost half a minute left. So chicken, cat, and goat, according to the question. The correct answer is I think a zoo or something. So 10 seconds left. Almost everybody is done with the question. 44 results. Yes, and only 18 were correct. So what was the correct answer? Who knows? Okay, so the first 3 letters from chicken is chi. The two from the second one is the CA, and GO is for altogether Chicago. Okay, so it was a town. So the last one is coming as far as I remember. Oh, this was the last one. Sorry. So surprisingly, Julia Kerestu is our winner. So congratulations to you and congratulations to all of the participants. I do hope that, and we do hope that it was not a really tricky one. So next time, if you use Kahoot, remember us, okay, for these tricky situations. So thank you very much for your participation. So we should go for our next step in our program.
And I have a list of duties, so I hope you are not that hungry, but I will try to keep it short. So the next point on our agenda is a very exciting point, because it was already mentioned, you were there last night, many of you, that we feel that we are a big family, and sometimes we mean this literally, because now let's welcome the youngsters, very young, two excellent dancers. I wanted to say scientists, but it will come later, who will give us very nice folk dance. They are called Teblab Art School, and we will see dances from Satmar region and dances from Galgament region. And the young boy is literally part of our family, so please welcome him. And the excellent pair of him. So it was the first wonderful performance from the Téblab Art School, Budapest, and we are waiting for the second one. Just relax a little bit. We enjoy a lot. Jó? Thank you very much. So, Gergő and Hanna, thank you very much. We really enjoyed. We hope that you could taste a little bit the Hungarian culture, not only through food, but through these very nice performances as well. So I have two more duties for tonight before we move forward and enjoy the evening together with the dinner. First of all, I would like to thank the organizers for the work they did, because you saw some of the people here, but many of the organizers were behind the scenes. So I would like to ask you guys, please come to the stage. I first, of course, would like to say thank you to Victoria Zsomuha, who you met through the emails and also here in person. Then László Baranyai, Laci. Laci, please come to the stage. Laci. Yes, he's still working, but please come to the stage. Thank you for the work. Timi Alkasab. Thank you. And the one who we learned from that we can further improve our digital competencies, Tamás Zsom. Katalin Kloss, Kati, please come from behind the scenes. Mercy, please come. And it would not work without IT. So Gábor, please. Gábor Horinka. So the close team of the organizing committee, thank you very much for all of your work. And of course, all of you who helped behind the scenes yesterday, today, and all the way. One of you asked me yesterday when we have started the organization of this conference. And, you know, I think we have this conference in every other year. So now from tomorrow, we start organizing the next. And I think you know that this is how it works. So thanks a lot for all of you. Thank you once again. And now my last duty for tonight is to close this conference. And I have a difficult task, to be honest, because I really enjoyed, I learned a lot. So I was thinking, getting to the end of this conference, what would be a proper closing of today? And I asked ChatGPT, so what do you mean? What do you think? What is a good conference? And what are the parameters we should consider? And it said that the conference is good if it has a clear purpose and relevance. And having the wonderful presentations today, I was listening, I saw a lot of connections. So I believe that we set a goal. We called our conference Biosystems and Food Engineering Conference. And I think that was a clear purpose, a clear goal, what we fulfilled, because we saw presentations from very diverse fields of agriculture, food science, and we started talking in a broader view about biomass, how we need to combine our forces and continue working, doing research together. And then ChatGPT told that a conference is good if high quality content is provided. Now, I believe we also fulfill this requirement, because just from the first presentation, hearing from the beauties of 3D printing, then the advanced digital technologies through different presentations, We also learned that the biogas production does not necessarily come with smell, bad smell. Then we also learned that we have to be careful how much we sweating, because this can tell us a lot about ourselves and about our behavior. And the very important thing I learned that waste should not be considered anymore as a problem, but more as a solution and a valuable component for the future. So I myself also learned a lot. I hope you also learned from each other. And I believe so, because I already saw you started chatting and continue discussions about related topics. Now, the third thing what ChatGPT mentioned is the effective organization. I let you to decide how you feel, if we fulfill with this question or not. And the last 2 points, and I'm merging a little bit because there was a very interesting list from ChatGPT: networking opportunity and diverse and inclusive participants. I believe this happens. So we had a very nice networking opportunity starting yesterday, having the wonderful coffee breaks discussions, and also the discussions after the presentations. And I also believe the participations are very diverse and inclusive. Just to mention that there were more than 100 participants here in person and online. And we are very proud that more than 30% of the participants are invited from abroad. If I count our own students who are also from abroad, then of course it's even more higher. So that is truly an international conference. And very important is the next generation. So I believe the diversity also came not from the different nations, but from the different seniority level. So more than half of the participants are students, PhD students, master students, and also bachelor students. So I think the conference has been successful from this point of view as well, because I think we should not be only good scientists, but also good teachers to teach the future generation, to show them ideas, what they can develop further. So I believe this could happen through these 17 oral presentations and 90 plus poster presentations. And the last thing what ChatGPT mentioned, I think it's the future. Post-conference impact. So it's on us, right? So I can't tell you today what will be the impact of this conference tomorrow and then after, but I believe we have again started something because we got to know each other. So as I tried to mention the last night as well, we feel that we want to bring together you, our partners, our friends, not only for us to get you in more details here, to get you know in more details, but also to connect the different fields. So let's collaborate together in the future as well. Let's continue what we have started. We are always here. So if you missed talking to someone, you need his or her contact, let us know, and let's continue doing research together, generate projects together, and create a better future for humanity. So thank you very much for all of your involvement, and let's enjoy the dinner together. Thank you. The technical announcement, the dinner starts at 6 in the neighbor room, so enjoy.